Welcome to part 2 of this video on the immune system. Okay, so far what we have discussed is the first line of defense, which is part of the innate immune system, and this consists of physical and chemical barriers to try and prevent pathogens from actually being able to get inside our bodies. What we're now going to talk about is another part of the innate immune system, which is known as the second line of defense. And this is still part of the innate immune system, so the mechanisms are non-specific. And by the way, just going back to the first line of defense, hopefully you'll agree that all of these mechanisms are very non-specific, so indeed they should be grouped under the innate immune system. Now we're going to move on to the second line of defense, and this is also going to be a non-specific mechanism. We're going to use the same mechanisms no matter what pathogen we're actually dealing with here, and it's actually called the acute inflammatory response. So more commonly than being called the second line of defense, we often call it the acute inflammatory response. Okay, so let's suppose then that we have some pathogen which has managed to breach through the first line of defense. So it's got through the epithelium and it's now in the actual tissue. So let's just draw this here. So let's say this is our pathogen, and this is the picture that I'm going to be drawing throughout this video just to illustrate a pathogen, a red blob here. So this is our pathogen and it's now in our tissues. Okay, so what are we actually going to do about this now? Because this thing might be dividing and dividing and dividing now and producing a whole bunch of pathogens inside our tissues. Or indeed we might have managed to get lots of the pathogen in all at once. How are we actually going to deal with this? Well, the major troops, if you like, that can deal with pathogens, they are in the blood. So there are cells in the blood, and there are also very powerful proteins in the blood that are put into the blood by the liver, which are capable of attacking pathogens. So what we want to do is we want to get those troops, those proteins and those cells capable of attacking pathogens from the bloodstream where they're just circulating inactive to this actual site where we've got the pathogen invading. So that's what the acute inflammatory response is all about, trying to mobilize the troops. It's a plea for help, basically, by the tissue here. So the first thing that needs to happen is we need to recognize that there is an actual pathogen present in the tissue. We then need to call for help, and we then need to actually get the troops in, and we need to understand what the troops are going to do once they're here. So that's the way in which we're going to approach this. So the first big question is, how do we actually recognize the presence of this pathogen? How does a tissue that is infected with a pathogen recognize that it is infected with a pathogen? Well, there are a bunch of cells which are collectively known as sentinel cells that are going to be extremely important here. So sentinel cells are cells that you have all over the body. So no matter what tissue you go to in the body, you will find sentinel cells, and they're distributed around the normal cells of that tissue. So they're dotted around, if you like, and their job is to continuously be on the watch. They're continuously looking for signs of pathogens, and if they find signs of pathogens, they then set off the alarm. Okay, so sentinel cells. So let me give you some examples then of sentinel cells. So major examples of sentinel cells are dendritic cells, macrophages, and mast cells. So I want to draw a picture for each of these. So we'll start off with dendritic cells. So dendritic cells, the reason they're called dendritic cells is that they have dendrite-like processes. So they are similar, if you like, They look similar to the dendrites of neurons, but these cells are very different from neurons. They do not have the appropriate voltage-gated ion channels in order to conduct action potentials. But they do have these dendrites coming off them like so, hence why they're called dendritic cells. Okay, so sort of star shape like so, with these processes known as dendrites coming off the major cell body in the center. So I'll just colour this cell in. So you have dendritic cells all over the body, so in different tissues all over the body you'll have these cells, and they're just sort of interdispersed amongst the other cells of that tissue. And you can imagine that these dendritic processes, they're going to stretch off through the tissue. And this is why this cell has these dendritic processes. It's so that this one cell can feel a huge amount of, a huge volume of the tissue, and it can be on the lookout for pathogens in that large volume of tissue. So it extends these dendrites into the surrounding tissue. So if you like, here is a more normal cell for the tissue. So these are the parenchymal cells here, and the dendritic processes, you can see, are extending into the tissue, and they'll just be continuously feeling around for signs of pathogens, and if they find signs of pathogens, then they'll set off the alarm. So dendritic cells are one of the major cell types that's going to be looking for pathogens. Okay, right. Another major type of sentinel cell then are resident macrophages. So macrophages are a type of cell of the immune system that we're going to recruit hugely as part of the acute inflammatory response. However, tissues of the body do have macrophages already present within them, and these are known as resident macrophages, and these are working as sentinel cells. So all over the body, just like dendritic cells, you have resident macrophages dotted around tissues, and again, these are on the lookout for the presence of pathogens, and if they find them, they're going to set off the acute inflammatory response. And indeed, as part of the acute inflammatory response, we'll bring in loads more pathogens, which are going to be one of the major troops to destroy the we're going to bring in loads more macrophages, which are going to be one of the major troops that will help destroy the pathogen. Okay, so let me draw a picture of a resident macrophage. So macrophages are great big blobs of cells. So it's quite easy to draw a macrophage. They just look like so. So here's the nucleus, surrounded great big blob of cytoplasm. Okay, so here is our macrophage. So resident macrophages because they're resident within the tissue. They live there. Okay, and the final major type of sentinel cell that I want to mention are the mast cells. And these, compared to these two types of cells, are much smaller. So they're a much more reasonable looking cell here. I'll just sort of move this up a little bit. Here is the nucleus of the mast cell, and one of the key things that mast cells have is they'll have granules, so they'll have little vesicles which are full of histamine. So I'll just colour the mast cell in here, and I'll colour its vesicles specifically in there. So those are the granules of the mast cell there in green, and they're going to be full of histamine, stored histamine, and histamine is going to be one of the key alarm bells that the mast cells can release to call for the acute inflammatory response to begin. So they've got granules, and in fact I'll label those up as histamine granules. Okay, so they're full of histamine molecules, which is a very small molecule. You know, a molecule that you could draw its structure out very easily. It's the amino acid histidine with the carboxylic acid group removed. Okay, right. So these are our three major types of sentinel cells then: dendritic cells, resident macrophages, and mast cells. And as I say, they're dotted around tissues all over the body, and their job is to continuously look out for signs that pathogens are present, and if they find evidence of the presence of pathogens, they are going to release all sorts of molecules which are effectively pleas for help, screams for help, or alarm signals, if you like. Okay, so the next question then is, how do they recognize the presence of a pathogen? How do they know whether there is a pathogen present in the tissue that they are present in? Well, what they have is receptors for molecules that indicate that there is a problem. And these are molecules that should not be in the extracellular fluid. So you know, these are in the tissue, and they continuously are feeling the extracellular fluid. They have receptors on their surface, and these receptors will be feeling the extracellular fluid. They'll be looking for signs in the extracellular fluid that there is something wrong. Now, normally there are loads of different molecules in the extracellular fluid, and these would not be activating these receptors. However, if you have a pathogen present in the tissue, then all sorts of molecules will be present in the extracellular fluid that no human cell would ever make in its right mind. So these are a giveaway sign that there is a pathogen present, and these are the sort of things that these sentinel cells are going to recognize and are going to then set off alarm signals for. So there is an umbrella term to cover all of these different molecules that pathogens can produce and which will end up in the extracellular fluid if a pathogen is present in the tissue, which no human cell would ever produce in its right mind. And these are known as PAMPs. Now this stands for something. So the first P is for pathogen. The A is then for associated, so pathogen associated. And then the MP is for molecular patterns. So pathogen associated molecular patterns. So this is the umbrella term encompassing all of these different molecules that different pathogens might be able to produce that are a giveaway sign that there is something non-human present within the tissue. They're molecules that no human cell would ever produce in its right mind. So these are giveaway signs that these sentinel cells can have receptors for, and if the receptors get their ligand, they will activate intracellular cascades to cause these cells to release their alarm signal cells. Another important concept is the concept of DAMPs. So this is a related concept. This stands for damage-associated molecular patterns. So rather than pathogen-associated molecular patterns, this time it's damage-associated molecular patterns. Now these are molecules that might be present in the extracellular fluid that would indicate cell damage rather than necessarily the presence of a pathogen. So if you've had cells in this tissue dying, potentially because of the pathogen or potentially because of injury for some other reason, then this can also trigger off an acute inflammatory response. And the reason is that loads of molecules will be released from the inside of the cells that have been damaged and broken open potentially into the extracellular fluid. And these molecules would never normally be in the extracellular fluid, and these can activate the sentinel cells as well. So these molecules that are released when cells are damaged or dying, these are clustered under the umbrella term DAMPs, and they can activate sentinel cells to activate the acute inflammatory response as well. So the archetypal example of a damage-associated molecular pattern would be ATP, the major biological energy currency. Now that's a major molecule that should be in the side of cells, but it should never be in the extracellular fluid, and if it is in the extracellular fluid, it shows that cells have been breaking open and releasing that ATP into the extracellular fluid. So it's a giveaway sign that you've had cell damage. Okay, so the sentinel cells then, they are going to have receptors on their surface for pathogen-associated molecular patterns and damage-associated molecular patterns. And there is another umbrella term for these receptors. So if I draw one of these receptors on here, the umbrella term for the receptors that can recognize pathogen-associated molecular patterns and damage-associated molecular patterns is PRRs, and this stands for pattern recognition receptors. So pattern is the P, the first R is for recognition, and then the final R is for receptors. So again, this is the umbrella term that encompasses all of a huge number of different receptors for specific pathogen-associated molecular patterns and specific damage-associated molecular patterns, which recognize these categories of molecules that can then set off the acute inflammatory response. So what I want to do, because all of this is very generalized, PAMPs, DAMPs, PRRs, I want to give you a specific example of a PAMP and the pattern recognition receptor that it has. So a nice example then, the archetypal example that all textbooks use, is the example of a lipopolysaccharide, LPS. So this is a molecule that's produced by gram-negative bacteria and which is used in the outer membrane of gram-negative bacteria. So if I just draw a little picture of a gram-negative bacterium. So gram-negative bacteria, remember that those have two cell membranes. Well, in fact, sorry, they have two membranes. They don't have two cell membranes. The inner membrane is considered the cell membrane. They have two membranes, like so. So the inner membrane here is considered the cell membrane, and then this is the outer membrane. So you shouldn't, as I just did say, you shouldn't say that they have two cell membranes, because it's only the inner membrane that's considered the cell membrane. And then the space in between the inner and outer membranes, that's called the periplasmic space, and that contains the cell wall of the gram-negative bacterium. So this is the periplasmic space, and I'll just put in here, coloured in orange, the cell wall of this gram-negative bacterium. So that's the periplasmic space containing the cell wall. Okay, now the outer membrane of the gram-negative bacterium, so I'll just label this up actually. This is a gram-negative bacterium. That means that if you stain it with the gram staining process, it shows up red, and it implies that the bacterium has this structure. So it's a gram-negative bacterium, and this type of bacterium, they tends to have this molecule known as Lipopolysaccharide as one of the components of their outer membrane. So it's one of the molecules that they have making up their outer membrane. And this is a molecule that no human cell would ever, in its right mind, use in its cell membrane. Now, if you've got one of these gram-negative bacterial cells in your tissue, then lipopolysaccharide molecules will be in the extracellular fluid, because, you know, the membrane isn't completely stable. Occasionally, some of the LPS molecules will break free of the membrane and end up in the extracellular fluid. And this is a giveaway sign that gram-negative bacteria are present in the extracellular fluid, and indeed we have a pattern recognition receptor for LPS, and the specific name of that pattern recognition receptor is the toll-like receptor 4, TLR4. So these pathogen-associated molecular patterns, they have pattern recognition receptors which are then going to activate the sentinel cells. So toll-like receptor 4 is the specific pattern recognition receptor for the PAMP LPS. So this is toll-like receptor 4. So why have I given you this specific example? It was just to make concrete this concept that different pathogen-associated molecular patterns and different damage-associated molecular patterns will have their own unique pattern recognition receptors. Because from this concept, from these words, it doesn't necessarily imply that. You might be thinking, well, is there just one pattern recognition receptor which can recognize all of them? No. The different ligands here, the different PAMPs and DAMPs, they'll have their own pattern recognition receptors. This is just the umbrella term for all of these different receptors that can recognize different PAMPs and DAMPs. Okay, so now the principle is that once you've activated a pattern recognition receptor on the surface of one of these sentinel cells, it will then activate the sentinel cell to release all sorts of alarm molecules that are then going to call for help. Okay, so let's go over onto the next page then, and we'll discuss these alarm signals. So there is a fancy term for these alarm signals. Rather than calling them alarm signals, immunologists prefer the term pro-inflammatory cytokines. I'll come to cytokines. Pro-inflammatory mediators, slightly more generalized than pro-inflammatory cytokines. Okay, so these sentinel cells are going to release a variety of molecules, a huge number of molecules in fact. And again, we've got an umbrella term for all of these different molecules. They're all grouped under the term pro-inflammatory mediators or pro-inflammatory molecules. Okay, so I'm not going to list absolutely every single one of the pro-inflammatory mediators that can be released by sentinel cells once they've been activated to do so by the presence of PAMPs or DAMPs. I'm going to give you a list, however, a short list. I'm going to give you in total 7 different pro-inflammatory mediators. In fact, fewer, 6 different pro-inflammatory mediators, and I'm going to describe what each of them does. I'm going to give you the major ones. There are loads more that you can go away and learn if you are so inclined, but I'm going to give you the major ones. So, pro-inflammatory mediators that are going to be released by the sentinel cells once they've been activated to do so. So one of them we've met already. It is histamine. Histamine is specifically going to be released by mast cells over the other types of sentinel cells. So histamine is one of the pro-inflammatory mediators that's going to be released by mast cells, and this is an extremely quick-acting one. So we're going to be discussing histamine first because it's one of the ones that acts quickest, and then the others will act after histamine. So histamine is a major pro-inflammatory mediator. Okay. Next up, we have three molecules that I'm going to group together as the pro-inflammatory cytokines, and I will define in just a moment what is meant by a cytokine after I've given you their names. So I'm going to give you the major ones. I could put so many in this great big list, but I'm going to give you the major ones, the ones that you really have to know the names of. But you could put loads more minor ones onto this list. So the absolutely major ones are tumor necrosis factor alpha and interleukin-1, although I'm going to add another one on, which is interleukin-6. If you're just going to learn two pro-inflammatory mediators, the two that you must learn are tumor necrosis factor alpha and interleukin-1. They are the major ones. They're the most important ones that you absolutely have to know the names of. I'm also going to discuss interleukin-6 because I find it an interesting one. Okay, and the action of interleukin-6 is well defined and well understood. Okay, so let me just write their full names out. So TNF-alpha here, this stands for tumor necrosis factor alpha. Okay, and there are other tumor necrosis factors, although this is the most important tumor necrosis factor. So tumor necrosis factor alpha. IL-1 and IL-6, they stand for interleukin-1 and interleukin-6. So the IL here stands for interleukin, between leukocytes, which is a fancy word for white blood cells. So this is interleukin-1 here. Okay, so collectively these three molecules, which are proteins by the way, they are known as pro-inflammatory cytokines, and as I say, you could add a huge number of other proteins onto this list of pro-inflammatory cytokines that are released by sentinel cells, but these are the absolutely major ones. And if you just want to learn two rather than three, the two to learn are tumor necrosis factor alpha and interleukin-1. Absolutely essential that you know their names. Pro-inflammatory cytokines. Okay, so pro-inflammatory, that's obvious, that just means pro-inflammatory. It means it's going to cause the inflammatory response. It's going to drive the inflammatory response, the opposite to anti-inflammatory. Cytokines. What is meant by the term cytokines? A cytokine is a molecule that is released by an immune cell, and specifically it has to be a protein. That's important. So you wouldn't describe histamine as a pro-inflammatory cytokine because it's not a protein. So cytokines are proteins, that's one of the key words, that are released by immune cells. So they're released by some sort of cell of the immune system, which all of these sentinel cells are. So dendritic cells, resident macrophages and mast cells, they are all counted as part of the immune system. So they're released by immune cells, and they are going to have an effect on another cell. So they're released by immune cells, and they're going to change the behavior of some other cell. So it's just basically a protein signaling molecule that is released by an immune cell. That's all. And the cell that it takes effect on does not necessarily have to be an immune cell. It can just be a general cell of the body, but the cell that releases it does have to be an immune cell. Otherwise, we're just talking about a hormone, basically. Okay, so that's the definition of a pro-inflammatory cytokine. So that's why I can't put histamine under this category, because histamine is released by immune cells. It does take effect on other cells, but it's not a protein, so it isn't a pro-inflammatory cytokine. So that's why I've had to have this broader title of pro-inflammatory mediators, which are just molecules now that have a pro-inflammatory effect, and histamine is certainly a molecule. Okay, so we've got 4 of them now. I want to do 2 more. So the next 2 are the lipid mediators, the prostaglandins. So they're also going to release prostaglandin I2 and prostaglandin E2, and I will discuss what both of these do later on. Okay, so just to write out their full names then, so PGI2 and PGE2. So the PG stands for prostaglandin. So this is prostaglandin I2, this is prostaglandin E2. Prostaglandin I2 also has another name that is still sometimes used, so I'll give it to you, and it's also called prostacyclin. So if you hear people talking about prostacyclin, that's just another name for prostaglandin I2. So So these are not proteins, they are derived from the lipid molecule arachidonic acid. So they're sometimes called the lipid mediators of inflammation, but I'll just refer to them as the prostaglandins. So we've got our small molecule mediator, which is histamine, we've got our pro-inflammatory cytokines here, and we've also got our prostaglandins. And this is my complete list of the pro-inflammatory mediators. As I say, if you go on to other sources, you can get much longer lists of the pro-inflammatory mediators released by sentinel cells, but then they don't tell you what the different ones do, so I kind of feel it's hardly worth actually knowing about them in that case. Whereas I'm going to explain what each and every one of these does, so we'll build it into a nice cohesive picture. So these are the ones that I think it's worth knowing about, and I'm going to explain exactly what they do. Okay, so where have we got to so far? So let's just summarise where we've got to. So we've seen the recognition of the pathogen present within our tissue by the sentinel cells. The sentinel cells have now recognised the pathogen is there, and they have now been activated to release all sorts of molecules. Mast cells release histamine and then all of these other molecules as well. So tumor necrosis factor alpha, interleukin 1, interleukin 6, prostaglandin I2 and prostaglandin E2. The other sentinel cells, the dendritic cells and the macrophages, they will release all of these apart from histamine. So histamine is just the mast cells, but they'll release all of the others. So these other 5, they're released by all the sentinel cells. Histamine, the top one, is just released by the mast cells. What we now want to see then is, what are these molecules going to do? Well, remember what we're trying to do. Remember our big picture of the acute inflammatory response. We are trying to recruit troops from the blood into the tissue. How are we going to get the troops out of the blood into the tissue? Well, we have to have an effect on the microvasculature. The acute inflammatory response is fundamentally a response of the microvasculature to these pro-inflammatory mediators. It's a change in the vasculature. So what I now want to talk about, before we actually talk about what the changes are going to be, is just a reminder of the microvasculature, because it's so important to understand this in order to understand the pro-inflammatory mediators and what they're going to do to the microvasculature. So for now, it's just going to be a little bit of revision, hopefully, of the microvasculature. Okay, so all tissues of the body have microvasculature blood vessels which are supplying them with glucose and oxygen and other nutrients. And this microvasculature is where the acute inflammatory response is actually going to occur. Of course, it has to occur there because that's where we have the opportunity to bring stuff from the blood into the tissue. So the microvasculature then, it consists of the arterioles, the capillaries, and then the venules. So just drawing a basic picture of this then, what we're going to start off with is here, let's let this be the arteriole that is just before the capillary bed. Now, the term arteriole, or rather the word arteriole, this is used to describe a huge different array of sizes of blood vessels. So it's quite a bad term, because when someone says arteriole, it could be a huge number of different sized blood vessels. There are very large arterioles that are almost visible, and then there are these tiny little arterioles that I'm talking about here, which is an arteriole just before we split into capillaries. So if you really want to stress the fact that you're talking about an arteriole that is absolutely tiny, the arteriole just before we split into capillaries, you can put the word terminal in front of arteriole. So a terminal arteriole is an arteriole that is just before we split into capillaries. So it's one of these tiny arterioles, rather than one of the arterioles that's only just smaller than an artery. Okay, so that's just dealing with the fact that this term covers a huge, great spectrum of different sizes of blood vessel. Okay, so now what's going to happen is our terminal arteriole here is going to split into even smaller blood vessels, which are the capillaries, which are one cell thick. So the lumen of a capillary can just about fit one red blood cell in it, like so. And then here they are reconverging up into a venule. And again, the term venule is a bad term because it covers a huge spectrum of different sized blood vessels. So if you want to stress the fact that you are dealing with a venule just after the capillary bed, you can put the term post-capillary venule there, and that gets across to people the fact that we're dealing with an absolutely tiny little blood vessel here, the venule just after the capillary bed. So that's the post-capillary venule. Here's the terminal arteriole, and here in the middle, these are our capillaries. Right, and I'll colour in the capillaries in yellow here, like so. So what I now want to do is just take cross-sections of each of these blood vessels and show you what the structure actually is. And before actually I just do that, let me just stress that all tissues of your body will have these present in them. They'll have loads of terminal arterioles splitting into capillary beds, and then the capillary beds will reconverge up to post-capillary venules, and that's where gas exchange and glucose delivery will occur at the capillaries. So all tissues have these structures within them. Right, so now what I want to do is just take cross-sections of each of these blood vessels. So I want to cut across a terminal arteriole, cut across a capillary, cut across a post-capillary venule, and show you the structure of the blood vessel wall, because this is what I really need you to understand, so that you can understand the acute inflammatory response. So we'll start off with the terminal arteriole. So I'll draw this, where should I draw it? I'll draw it up here. So we'll start from the inside and work our way outwards. So the innermost portion then of the arteriole wall consists of the endothelial cells sitting on a basement membrane. So here, these can represent my endothelial cells, and I'll just give them nuclei like so. So these are the endothelial cells lining the lumen. So this is the lumen of the blood vessel here. And then I'll put a basement membrane, as always. Remember, endothelia and epithelia, they're always on a basement membrane. This is what is keeping the whole thing together. You might ask, well, why don't the endothelial cells just fall into the lumen here? Well, they are attached to the basement membrane. Okay, so in orange there, that's representing the basement membrane. Now I'll just add a few arrows on here. So I'll put BM for basement membrane, and these are endothelial cells, so I'll put ECs for endothelial cells. Right, and then surrounding that layer, you then have a layer of vascular smooth muscle cells, and this is the key difference between the wall of the terminal arteriole and the wall of the capillaries and post-capillary venules, that we have something outside of the endothelium here. So this now is a layer of vascular smooth muscle cells, and for short I'll abbreviate vascular smooth muscle cells down to VSMC. So this stands for vascular, S for smooth, M for muscle, and then C for cell, and then obviously the S just to make it plural. Okay, so this will consist of lots of vascular smooth muscle cells, which are spindle-shaped cells. So I'll just draw one in, like so. So they have a thick belly and thin ends, that's what spindle-shaped means, and they will be arranged in circles, rings. So you'll have another one here, another one here, and the ring will continue on, so you'll go all the way around the diameter of the blood vessel here. Now, what this means is that the vascular smooth muscle cells can actually control the diameter of the arteriole, because if you imagine what will happen if all of the vascular smooth muscle cells in a ring like this were to contract, what would happen? Well, they'd all shorten in length, and then if they all shortened in length, that would mean that the circumference of the ring would shorten, and when the circumference of a ring shortens, I'll try and show this, if I reduce the circumference of a ring, what also happens? The diameter of the lumen is going to reduce. So what you can do then is vasoconstrict the arteriole, and contrary, if they were all to relax down, that would increase the diameter of the lumen of the blood vessel, so vasodilation. So vascular smooth muscle cells, they are responsible for controlling how wide the lumen of the arteriole is, and that controls how much blood is actually going to be flowing through this capillary bed. If this is wide, you'll get more blood flowing through the capillary bed. If this is narrow, then you will get less blood flowing through the capillary bed. So arterioles, terminal arterioles, control how much blood is going to be flowing through the capillary bed, i.e. how much blood is going to be flowing through the tissue. Okay, so that's wall structure of a terminal arteriole. Let's now do the wall structure of a capillary. So if I was to draw this in scale with the size that I have drawn my terminal arteriole here, the capillary would be something like this size here. But what I want to do is zoom in a little bit more, because you're not gonna be able to see that with the camera, especially under this light. So I'm gonna zoom in, but this means that it won't be in scale with the size of the terminal arteriole here. So here it is now zoomed in on, and it is a tiny little blood vessel, a capillary. It really can be completely surrounded by a single endothelial cell, like so. So here's just one endothelial cell making up the entire endothelial diameter of this capillary here, and it'll just be sat on a basement membrane. So here is the basement membrane that this endothelial cell sat on, and that's it. That's the wall of a capillary. It's just an endothelial cell sat on a basement membrane. So going back to putting it in scale, this would be the basement membrane, and then you'd have an endothelial cell on that basement membrane. Okay, so as I said, it's one cell thick, so a single red blood cell can fit through this lumen. So that's the vessel wall structure of a capillary then, and this is of course the site of gas exchange and glucose delivery. Now, let's go on to the postcapillary venules. So, the postcapillary venules are basically just like larger versions of capillaries. They have the same wall structure. Their wall literally just consists of an endothelium. They don't have a vascular smooth muscle cell there outside of that. So they're going to look like a terminal arteriole, but without the vascular smooth muscle cell there. So this is in scale now with this picture again over here. So here is the basement membrane in orange here. And then again we'll have endothelial cells sat on the basement membrane, like so. And I'll just give them all nuclei. But then you won't have anything outside of that, so that's the entire wall thickness now covered. So that's what a postcapillary venule looks like, this venule that's immediately after the capillaries have reconverged. So you go from that, to that, to that. Okay, so we now know the wall structure of these different components of the microvasculature, and this will stand us in good stead for understanding the acute inflammatory response, because the acute inflammatory response is going to be totally and utterly a response of the microvasculature in the tissue. Because after all, remember the big picture of what we're trying to do. We're trying to bring in troops from the bloodstream. These vessels control what can come out of the bloodstream into the tissue, so we need to change the behavior of these vessels in order to actually get the troops being delivered from the blood to the tissue. Okay, so we'll have a break here, and in the next video we'll actually look at how these pro-inflammatory mediators, which at the moment we've just seen their names, are actually going to affect these components of the microvasculature.